Okay, so I think I have just about everyone in, so I'll start sharing my screen. Hopefully it goes a little bit smoother than last time, but just bear with me. Oh my goodness. One of these days, guys, I'll get this figured out, I promise. Can everyone see my screen okay? I can't see the chat, so you'll have to verbally reply.
Yeah, looks good.
Yeah, we can see it.
Okay, so this is lab 5. We're going over mark, recapture, and population size estimates. So first off, what is a population? Does anyone have a general idea of what the definition might be?
Just like a bunch of individuals living like in a same area of the same species.
Yeah, so you just about nailed it. So it is a group of individuals of the same species inhabiting a given area at a given time. However, there are kind of 3 main problems with this definition. So how do you decide what a given area is? The ecology of a system is dependent on the spatial scale at which it's being studied. What constitutes an individual? Are they unitary or are they modular? And then what constitutes a species? So you can have various species concepts. So to kind of go over through the examples of this, the first image you guys can see, that's Trembling Aspen. So many Trembling Aspen, the plants are modular, meaning that you have one single organism that has multiple clones of itself, which are still attached through a main root system to the original plant. So meaning that it's not several different individuals, it's actually one individual that's just taking up a whole area. And then just an example of this, this stand of Trembling Aspen in Washington is considered by some to be one of the largest organisms on Earth. So the next issue with the equation, or with this statement, is with the Ambistroma laterale. This is the blue-spotted salamander, and it represents an organism where the species boundaries are not super well defined. They hybridize with a number of other mole salamanders, and they actually steal portions of the genome and incorporate it into their own. So you kind of have this confusion of what's one species versus another, or if it's a hybrid. You can't really tell that by just looking at it. And then the last example is a lock. So defining the given area using terms that are relevant to humans may not actually be biologically relevant. So our understanding of how organisms interact with the landscape may not be accurate to the way that that population is actually determined. So how do we determine population size? There are 3 main sampling methods. You have direct census, quadrats, and transects, and then you kind of mark recapture. So going through the 3, the most straightforward method of figuring out population size is by direct census. So this is once you've figured out what the given area is, you actually go out and you count the exact number of individuals of your species in that area. So this is possible for some organisms, and it's the method that we usually use for figuring out human population, but it has very obvious limitations in any number of biological systems. So generally, it's not possible to go out and find and then capture every individual animal in a population. And because of this, we kind of move toward estimating population sizes. So the first method of this is using quadrats. So quadrat theory works by randomly selecting a subsample of smaller areas from your larger one. And then you count the number of individuals within each quadrat. The population densities from each quadrat are then averaged and extrapolated to the whole area. The limitations are obvious for mobile organisms. They aren't really going to stay necessarily in that small sub area, but for sessile creatures like plants, it works quite well. It's important to note that transects are considered a type of quadrat, where basically you just take one of these squares and you stretch it out. In other cases, you use mark recapture to estimate population size. So for a given area, a subset of the population is marked and then you release it. And then in the subsequent visit, you capture however many animals again. And the proportion of marked to unmarked animals in the recapture event is to be representative of the proportion of the whole population. So you can calculate a theoretical population size estimate. And this is what we'll be talking about in today's lab. So how do you mark an animal? We did somewhat similar to this to last week with Steph, but they're not so much for tracking per se. It's more for identification. So for large vertebrate, tagging is often an option. You can have bird banding, as you can see here, where they put a tiny band around the leg. You can use ear tags for both large and small mammals, as you can see on this bear and on this small mouse. For invertebrates, tags can work as well, such as in the blue crab, or you can actually mark the organism with paint or nail polish, some sort of hardened colorant that will stick to the individual. Reptiles and amphibians are usually marked by clipping or filing. So Steph was talking about with her turtle identification, you actually kind of file the marginal scutes, and then you would count them to do the identification. For amphibians or other reptiles, you can use toe clipping. The other method is pit tagging. This is very similar to the kind of microchip tag that you'd put into your family pet. Each tag is inserted using a needle, and it kind of goes under the skin. And basically, each one has its own identification number. When you use a small reader, it will tell you the number. The biggest problem is losing tags. Birds can lose these leg bands. Ear tags can be removed or lost or unfortunately ripped off. Paint can chip off or fade. Toes can grow back. And if you were to mark a younger turtle, as it grows, it might have those files alter, or if you have predation efforts, it might kind of change how you would view the tagging. And then there are also concerns of harming or disturbing the animal. So as spoken about last week, you always want to follow ethical standards to make sure the animal is being the least invasive, but you also want to make sure that however you're tagging them isn't going to change their behavior, their natural behavior in any way, because it kind of defeats that purpose. So yeah. So we have a couple of different mark recapture techniques. Once you figured out however you're going to do your marking of the animals, you can conduct a basic study. The simplest model is the Lincoln-Peterson index. In this index, you have two capture events. You have your initial and then your recapture. So the initial is when all animals are marked that are caught, and then the recapture event in which you determine the proportion of marked animals in the second sample. This portion of animals marked in your second capture event is assumed to be the estimate of the proportions of your population. And this is how you estimate total population size. So this is the formula. Can anyone think of any downfalls to this index?
Most populations aren't closed like in the real world.
Yeah, so that's one of them. But the main thing here is that there's only two capture events. So you have your initial capture and your secondary capture, and you're using only those two to determine the whole population. So it's a really limited sample of the whole population. Depending on the species that you're looking at, this may not be very representative. So the next one you have to kind of increase this is the Schnabel Method. It's basically using the Lincoln-Peterson over multiple capture events. And you kind of use this when animals can be kind of difficult to catch. Therefore, the more capture events you have, the greater the likelihood that you're actually getting a good estimate of the total population. And you can calculate variance and therefore standard error of the estimate to get a good idea of how close you might actually be. So this is kind of the formula of that with all of the variables outlined here. So the main thing is that you have the total number of animals captured and marked in your first event. That's N1. And then NI is the total caught in each subsequent event. And they're kind of included. They're going to have multiple of this. With big MI, the total number of marked animals in the population, divided by little mi, which is the total recaptures in each event. So you're getting a much better idea of the whole population because you're not just doing two sampling events, you're doing several. So going over the assumptions that Noah was mentioning, the first one is a closed population. So you're assuming that there are no births, there's no deaths, there's no animals coming into the population, and there's no individuals leaving. The second is that marked and unmarked animals are captured randomly. And the third assumption is that the marks that you're using to identify these animals are neither lost nor are they overlooked. Can anyone think of some issues with these? And then how time could play in like a role in violating any of these three assumptions? No? Okay.
Sorry, what was your question again?
So can you say, is this a realistic model for most animal populations? And does time play a role in violating any of the assumptions?
Oh yeah, I think it's unrealistic. And the time would definitely violate it just off the first point of the assumptions of closed population alone. To assume that nobody is born or die, first of all, doesn't work. And then emigration over time, your population will just kind of crash with it.
Yeah, exactly. So it's pretty unrealistic that there's going to be no births or deaths within a population. If the habitat for some reason isn't very good, you might have individuals leaving it to go find better habitat. Or if the habitat's really good, you could have new individuals, depending on the species, trying to move in. And then marks neither being lost or overlooked. Losing markings is very common. Ear tags, they could rip off, they could fall out. Even, you have to take into consideration this for the type of animal that you're looking at. So for the ear tag, a small mouse, its life cycle is much, much shorter than that of a bear. So if you're, depending on the type of study that you're looking at, you have to take in consideration the lifespan of that animal. And if that tag is going to hold up to the lifespan, if that's how long you want to study for. So lab exercise you guys are going to be doing, we'll kind of go through it in a document, but you're going to be using popcorn to kind of do that Schnabel mark recapture technique event. What you guys are going to be doing is you're going to run the lab exercise on your own during Zoom. Then you're going, we'll be in our breakout rooms. Then you're going to come back, you're going to answer your questions, and then we're going to discuss the responses in our own Zoom breakout rooms. So you guys can look over that Word document in detail. I'll kind of skim over it. And then you guys will also work on the Brown and King Solver population size worksheet. And then just to reiterate the whole point of doing our writings. So this is to build up to your final scientific manuscript. The key components here, you have an abstract, the introduction that you should hopefully be finishing writing, if not finished, to be handing in today, the materials and methods from last week. Today, we're going to be doing the results and discussion that you guys will hand in later, and then your lit cited. So the assignment you guys are going to be doing, you're going to write a results and discussion sections, and you're going to look at a study whose methodology uses mark recapture techniques. So you're not necessarily going to write about the exact techniques per se. You can write about population size, a measure of abundance, of your popcorn kernels, for example, in some scenario that you create. So for example, you could write about variation in kernel abundance among microwave popcorn brands, with the brand as your study location. You can even write your own or create your own animal if you don't want to use popcorn kernels. But basically be creative and have fun. For your results, again, you guys had your first table from a couple weeks ago and you had the option to do a bonus. Keep in mind, you need to do proper captions. You need to have row headings, proper line formatting, and displaying your summarized data. You can do a statistical comparison. It is optional. You can do a simple chi-square analysis to see abundance estimates. You don't necessarily have to, but I recommend it just because it's good practice. In the results, you include the summary of findings in text that are not redundant with the data in the table. So basically, you're not just restating in paragraph form what's in your tables. It's a little bit more complex than that. And you can refer to the table in text. And then in your discussion, you're kind of going from narrow to broad. This is where you interpret your data. You are not interpreting any of your data in the results. Your results section is simply just stating the biggest parts of your results to just make them pretty clear, but you're not going to think, oh, why is this way? What does this mean? This is where you bring it into your discussion. It's in a different section. You're going to compare your findings to those in literature. You need to cite your papers. And then your conclusion, take-home message, and future directions of study. This is in further detail in the handout. That's just very, very basic. The format, you guys are going to be doing it in 12's New Roman 12-point font. I recommend actually just changing the default in Word if you have it to be Times New Roman. That way you aren't automatically defaulted to Calibri or anything like that. It has to be Times New Roman. Just set the default. It makes your life a lot easier. You don't have to think about it anymore. It's going to be double spaced. You have to have your name and student number as a header. You're not doing a title page, but you do need to include a working title for your mock manuscript. So titling it Results and Discussion or Lab 5 is not sufficient. You need to have a working title that's more than 2 or 3 words. It needs to be a full title. You need to have at least 6 references from primary literature. So remember, these are things like journals or books, chapters from books, but they aren't textbooks. Basically, they need to be peer reviewed. Google Scholar is a great place to start. It says here to hand in as an email attachment to your TA or upload in D2L. I personally, for my group, I prefer it on D2L and then email it to me as a backup, just because I get a lot of emails. It might kind of get lost. So put it in D2L first. And if you want to email a version just to be safe, go ahead. And it's due in 2 weeks on November 9th. So I'm going to bring up the document. So can everyone see the Word document okay?
Yep.
Yeah, okay. So this is very much a kind of more in-depth review of what I just talked about. I recommend reading it just so you kind of get the full story. But basically, you guys are going to be getting basic concepts on population ecology. You're going to need an unknown quantity of popped popcorn in an opaque container or paper bag so you can't see through it. A bag of microwave popcorn that has about 460 kernels would be perfect. If you can do 3, that'd be even better. Since we are doing kind of 3 trial type, we're doing 3 different scenarios. So the more popcorn you have, the better. And you're going to need felt tip markers or pens in 3 different colors. So I'll very briefly go over the experimental procedure. Your GTA and each of your breakout rooms can kind of go through it more in depth if you have more questions. But the first, we're going to be doing an unbiased mark recapture. Basically, your popcorn is representing an animal population, and the paper bag is your study site. You'll be the sampler, the counter, and the data recorder. So that means, sorry, scroll down. There are tables in here. So you guys are going to fill out these tables on your own. There's no shared Google Doc like in the past. So you need to keep track of your own information. And you have to maintain a constant sampling effort, and you have to think of why that's important. Basically, you're going to do 10 trials. Each trial, you're going to reach into the bag without looking and grab a handful of popcorn. You're going to mark all the popcorn kernels captured by drawing a dot on each one using one color of your markers. You're going to throw it back in the bag, shake the bag, and then repeat the sampling for 10 times, as I said. And you're going to mark any new captured individuals in each trial. See table 2, that's where you're going to have your data table, and you're going to record the sampling trials from 1 to 10. The number captured, both marked and unmarked, combined, the number of new kernels, which is your new captures. For each trial. After your 10 trials, you're going to count the total number of popcorn kernels in the bag so that you know the exact number. Then your next table is you're going to assume that individuals disperse evenly into the population after marking and release. You're going to use the same procedure as above, but you're going to mark the popcorn kernels with a different color so you won't be confused by the first set from the first part, or this is where your second bag will come in handy. In this part, you're not going to shake your bag between trials. You're going to capture your popcorn, you're going to mark it, and you're just going to throw it back in the bag and then grab a new handful. So you should preferentially recapture the already marked kernels, and you're going to record the data and then again count the actual number of popcorn kernels in your bag. The third one is the negative recapture bias, using the same procedure but with a third color, preferably a third bag of popcorn. And what you're going to do this time is you're going to bury your marked kernels to the bottom of the bag, and you're not going to shake, so that your reducing your probability of recapturing the marked kernels. Again, you're going to record it and then count the actual number in the bag. And then I'm just going to go through an example using the Schnabel method. So you had trial 1, you captured 6 individuals. Obviously, it's your first trial, so you don't have any recaptures. So your new captures are 6. You have 0 as your cumulative number. And then for trial 2, you captured 5. 2 of those were captured previously, so those are your recaptures, meaning that you have 3 new captures that were unmarked. And this is where you start bringing your cumulative number. So you have 6 from the new captures above, plus 0 to give you 6. And then your third trial, you have 7 in total. 3 of them were captured before, meaning you have 4 new captures. So then you have 3 plus 6 gives you 9. Trial 4, you have 6 in total that you caught. 4 were previously captured before, so you have 2 new captures. So then you have 4 plus 9 gives you 13. And these are the values you're going to bring in to your equation. This is an example of how it lays out. So you have 6 multiplied by 0, and then you have 5 multiplied by 6. You have 7 multiplied by 9, and then you have 6 multiplied by 13, all divided by the added number of the total recaptures. So you have 0 plus 2 plus 3 plus 4. So these are your tables. You have your number captured, your recaptures, your unmarked, and your previously captured. Again, it has all of the variables outlined here with their letter codes. You know what they are. And then the discussion questions, there are 4 main discussion questions. So once you guys have done your experiment and you've calculated your population size using the Schnabel method with the formula above, you're going to make calculations for all 3 experiments for the unbiased, positive bias, and negative bias. And you're going to compare the results. Did you find a difference in population size as a result of sampling, and explain? How does your sample size estimate from each of the 3 experiments compare to the actual population size? What sort of real-world situations did the positive and negative recapture bias experiments simulate? And in light of the above considerations, what suggestions can you offer that would increase the accuracy of mark recapture population size estimates in the real world? So these are the 4 discussion questions that we're going to be talking about in our breakout rooms. For your assignment, again, you're writing a result and discussion. I've gone over the basics of this, but please read through it for clarification. In terms of writing a results section, read through all of this, but the main point is that your result should not be just kind of a redundant summary of what was already in the tables or the figures. People can look at that, the readers will see that, they don't need it restated to them in paragraph form. What they need to know is the big takeaways of that. So is A greater than B without giving all the values? Is something linear over a certain range? What is the optimum pH? These are all examples. And you're going to want to connect results to one another. So basically, you want the data to speak for itself. And remember that your busy readers will be grateful for help, but they don't need you holding their hand through the whole thing. Please, Jack, you might have different input on this. I don't think you should really have any conclusions in your results. That's typically something that you leave for your discussion, because that's where you're interpreting your data, you're figuring out what it all means. Again, for your tables and figures, you have gone through this. You guys should be pretty comfortable with this for now. You need to have a caption that is a standalone from the rest of the paper, meaning that if someone was to look at your figure or your table, they should be able to read your caption and look at your table or figure and understand exactly what you're talking about without reading any other part of the paper. Writing table 1 mark recapture is not sufficient. They need more information than that. But also with that, your caption cannot speak completely for the table. So the table and the caption, or figure and caption, they need to kind of complement each other. So if you put all that information in the caption, but you don't include that pertinent information in the actual figure or table, a reader's going to get confused. And then as a little tidbit, when you're marking figures or tables, try to make it as concise and clear and as easy to read as possible. The human brain gives you about 3 to 5 seconds to assimilate all that information. If it takes longer than that, your reader's not going to look at it anymore. They're going to get bored. They're going to move on. So if you can present all of your information in a super clear way, the better. And then in terms of your discussion, this is called heart of your paper. This is where you figure out what your results mean. But you also need to kind of be careful not to kind of ramble and add in a bunch of fluff and unnecessary information. Be concise. We've said it a whole lot. Tell the full story, but only include what needs to be there. If you're doing a comparison, say you find a result and you find papers that are controversial, summarize them concisely. Don't ramble on and on and on. There's this really good How to Write with Style. I'm not going to read through it, but I will read the main summaries. I highly recommend it. It's a good read. But basically, the summaries that you guys are going to have is find a subject that you care about. Do not ramble on. Keep it simple. Keep it concise. Have the guts to cut information. If it's not going to help your paper, don't include it, even if it sounds good. If it's not going to help you out, don't waste letters on it. Keep what needs to be there. Make sure that you sound like yourself. Say what you mean. Be sure to pity the readers. They have a lot of reading to do. Make it clear and make it easy for them to read. And that's everything from me.
All right. Does anybody have any questions for Natalie before we start to load people up into the breakout rooms that, again, did not seem to remember where they were supposed to be allocated? Are you guys, does everybody have their popcorn? Are you ready to get popping and move over to your rooms?
All set.
All set, Noah? Awesome. What flavor of popcorn do you have, Noah?
Extra butter on the one I got.
Nice. It's going to be extra greasy.
Yeah.
Excellent. Okay. Then, again, as I said, the breakout rooms didn't work properly yet again, and I don't know why. And I have not bothered IT about it because I think they're utterly overwhelmed by other things. So yet again, I'm going to have to open all the rooms and then get people to tell me which room I need to send them to again. And I'm sorry about that, but it's what we got. So I'm going to open the rooms and then I'm going to, yeah. Okay.
I'll bring up the list again in case anybody forgets.
Yeah. Okay. So I should first send the TAs over to make sure they're in rooms. So Brianna, do you remember which room you were actually supposed to be? No, I can figure that out. You're room one.
I'm room one, but I can join here. There's a button.
Oh, you can send yourself over as a co-host? Awesome. Steph, can you do that? Can you see that?
I can see it under the More tab.
Okay, so Heather, you can send yourself over?
I think so. I'll try right now.
Give it a try, yeah. I'll be able to tell if she disappears off my list and the screen. Looks like it's not working.
I see a comment, Jackie. There's a chirping.
Oh, there she goes. Yeah. Do you know what that chirping is, Tom?
It sounds very much like some crickets and some frogs.
There are crickets singing right now in my leopard gecko tank. All right, Steph, can you send yourself over? Is it possible for you to do it?
I don't see a button, no. Like even under More, I don't have the option.
That's weird. Okay, I'll have to send you over to your own room then. How about that? All right, room 3. There goes Steph. Okay, now are all the GTAs sent over once Steph goes? I think so. Is Natalie over in her room? Yeah. Okay, here I go. Adam. Caleb.
Brianna.
Thank you. Adam LePage. Not answering. Alex Muiz. Whose room are you in?
They're saying it in chat.
Oh, I can't see the chat because my breakout room's covering everything.
Adam is in Steph's and Alex is in Nat's.
I'll follow the chat. I'll follow the chat. I was looking them up.
Okay, Alex is where? Alex, please.
Natalie.
Thank you. Allie, Lake Khalil.
Heather.
Heather. Amanda Smith.
Steph.
Amy. I think you're Steph as well.
Yep.
Yeah. See, I can move stuff out of the way here. Okay. Amy Meyer.
Nat.
Annalisa.
Heather.
Heather. Anish.
Heather.
Antonia.
Brianna.
Brianna. Awab.
Brianna as well, sorry.
That's okay. Benjamin Demare. See in the chat? I don't see him. Okay. Benjamin Hadnaka.
I'm with Natalie.
Thank you. Brooke.
Brianna.
Cameron.
Heather.
You sound tired, Cam. Caroline.
Anna.
Charlie Morrison.
Heather.
Shaylin Andrews.
Benjamin says he wasn't sent to a room.
Yeah, I haven't sent Benjamin Demare yet. I didn't hear where he said he needed to go.
Why is that? Hear me now.
Well, that was a bad connection, Ben. I'm sorry I didn't hear.
I feel like that might have been Darth Vader or Kylo Ren.
Okay.
Yeah,
And Kylo Ren's name was Ben. Kylo Ren's name was Ben, just for clarity.
yeah. Ben, maybe you could tell us in the chat. Your connection is really rough there. Oh, Brianna. Okay, got it. Thank you. All right. Sorry. Sherry Lynn Andrews. Not hearing.
Hello.
Sherry.
She wrote hello.
She wrote hello. Who's your GTA, Sherry Lynn? Got nothing. Okay. Chloe.
Steph.
Chloe has Steph. Awesome. Colin Marr.
Brianna.
The question, Sherry Lynn, was who your GTA is.
Daniel Nout. Nout?
It's just no.
No.
Sorry, I'm running around the house trying to make popcorn and stuff now.
Yeah, awesome.
Don't burn it. Don't burn it.
Where am I going again? Where am I going? Is it Brianna?
Are you Brianna? Dan? Okay.
I think.
All right. I'll send
the
you
person who was just presenting. Is that right? I think so.
Sherry Lynn is going to Heather. Jackie?
To Heather. Thank you. All right, Darwin.
Steph.
You. Emma Brown. There's so many Emmas.
Nat.
Emma Jessup.
Heather.
Heather. And Emma Risto.
Heather.
3 Emmas in a row. Henri Lavelle. Don't see Henri.
Brianna?
Brianna.
Yeah.
Amen. Natalie. Jackson Daly.
Steph?
Thank you. James Barrett.
Brianna.
Janine Stephen.
Heather.
Heather. Jenna McIntosh.
Steph.
Jessica Turcott.
Brianna?
Johnny Lamb.
Johnny Lamb, love that name. Heather?
Heather. Jordan Lynch.
Brianna.
Kaylee Lowen.
I'm with Natalie.
Thanks. Kennedy Sikora.
Natalie?
Kiana Valentin.
Natalie.
Lindsay Bertrand.
Natalie.
And then Lindsay Van Dam.
Stephanie?
Is Steph. Luis. Heather. Mark Visser.
Steph.
Hello, everybody. So we have 13 out of 16 people that are supposed to be here. So I guess we might as well just get started. So I'm gonna go over just some notes about marking your all your work from last week. So generally it was well done. There were some issues still with people and their tables. So especially with this being the third shot at doing tables, we're going to have to be a little less lenient. A lot of people improved greatly and you did your tables correctly the second time, but there were a couple people that still were really, really off the mark with how to format it properly. So make sure that's something you pay attention to, because I feel like it's going to be something that would be really easy to nitpick when this is your third opportunity to write a table. Another note that a lot of people seem to do, so your results don't go in your materials and methods. So a lot of people wrote, this is what I did, and on the first day I ate a chickpea salad, and on the second day I ate McDonald's, and on the third day I ate this. So those are part of your results and would normally be put in a table. Your materials and methods is purely just enough information that somebody can repeat your experiment with their own lunches, and they have just enough information that they can do that. You don't want any more and you don't want any less. So it's not really relevant what you ate for lunch that week. In terms of displaying equations, I don't know if it was gone over with you guys. I didn't dock marks for it, but I did give you examples of how you should display equations. And for those of you, I'm pretty sure I tried to write it in a note on every single one, but because we were using a modified equation, you should be citing the original equation and saying how and why we modified it for our purposes. Just a general thing. And another thing, one or two students did it and I didn't dock marks for it because it's your materials and methods, so it wouldn't necessarily come up, but just for definitely in your results and your conclusions, and definitely in your introductions, which I haven't had a chance to look at, and for your final manuscript, you need to put the scientific names of what you're looking at. So you can use the scientific name or you can use the common name, but you have to state both of them at least once. Well, you have to state both of them. If you're just using the scientific name, you can use just the scientific name, but just saying like wood turtle isn't sufficient. You need to put the scientific name at least once so that we know, because common names can change depending on where you are. So those are just some general notes about the marking from last week. Like I said, overall it was done very well. And I will be marking your introductions soon. And again, another point I meant to say, if you have any questions at all about how it was marked or potentially how you're going to be marked, I mean, that's still a little bit up in the air until I see, because I have to adjust it. Sometimes if everybody did one thing wrong, it's assumed that we didn't communicate it clearly enough. So we sometimes adjust exactly how we're marking. But if you have any questions about how you were marked, or you think I missed something or anything like that, send me an email, because I am human and I make mistakes. There was one student where I did make a mistake on their marking and I was able to give them more marks. So look over what I wrote. If anything doesn't make sense, I can try to clarify it for you, or it could be a mistake. So let me know if you have any questions or concerns about how things were marked. And with that, we can now go to our popcorn lab. So hopefully everybody has their popcorn all ready to go. I'm assuming you do. So I think how we're going to do it, just to keep everybody kind of engaged. I'm not entirely sure exactly how long it'll take people, and I guess it depends on how much popcorn you have, because you have to count the total. And if you have 3 different bags of popcorn, you'd have 3 different totals. But we will start by doing, as Natalie explained, the Unbiased Collection. So is there anybody who has any questions, comments, or concerns about that first part, the Unbiased Collection? So just as a reminder, you take your handful of popcorn, you mark them, you put them back in, and you shake the bag. Okay? And we can discuss what shaking the bag and everything means after we're done all of the 3 methods, I suppose. So yeah, if everybody has all their stuff ready, you can go and there's no questions, and you can go and do your unbiased. And I will be here. If you have any questions, I'm going to turn off my video and mute myself just so I'm not disruptive. And also, by the way, feel free when we're in our breakout rooms, if you want to turn on your cameras or your microphones, if you're comfortable with that. Because we're in a smaller group, so it shouldn't really slow everything down too much. But yeah, so I'll be here if you have any questions, but go forth and do your unbiased and come back with your data and we can talk about it a bit.